So thank you, Jane. So as Jane said, my name is Alexandre, and I'm a PhD student in biostatistics in public health at INRIA Bordeaux Sud-Ouest Center in France. And I'm supervised by Rodolphe Thiebaud and Mélanie Prague. And so first of all, I would like to thank the organizing committee to give me the opportunity to present this part of our work today. So which is a collaborative work between our team, INRIA system team, and Janssen pharmaceutical team. And so this work is about the modeling, the within-host modeling of the dynamic of the humoral response following a two-dose heterologous vaccine regimen against Ebola virus. And so first of all, I'm going to start with a brief introduction of the context. So since 2014, the world faced two major Ebola outbreaks. So in 2014, 2016 in West Africa, and in 2018, 2020 in Democratic Republic of Congo, with more than 10,000 and 2,000 deaths respectively. And so unlike COVID-19, it's important to mention that Ebola virus is characterized by a quite high rate, mortality rate ranging from 25 to 90%, so which led to the declaration of a public health emergency of the international concern by the WHO in July 2019. And moreover, since 2014, the development of several vaccine candidates against Ebola virus has been accelerated, leading in particular to the approval of the two-dose heterologous AD26 ZEBOV and MVA-BN philo vaccine regimens, in particular in the European Union, for the prevention of the Ebola virus disease since July 2020. And so consequently, over the years, several works have been conducted to evaluate the efficacy of this specific vaccine regimens. And that is specifically the case of the work I did by Pazin et al. In 2019, published in GVI, which studies the establishment of the long-term immunity response on 3 phase 1 trials performed by the EBOVA consortium using ODE mechanistic models. And so in those trials, subjects were randomized to receive either AD26 ZEBOV and MVA or vice versa, with an interval of either 28 or 56 days between the two vaccinations, which represents a lot of information. However, despite this great variety of data, in these trials, patients were only followed during 1 year after the first vaccination, which consequently limited the information learned on the long-term immunity. And so the objective of the work that I'm going to present was to develop and to apply a new methodology to validate and to update this model using additional data which could bring more information. And so, for example, in our case, we use data from 3 phase 2 trials. So about the data, so in this work, we focused only on patients receiving AD26, then MVA with an interval of 56 days between the two vaccinations. And so in this work, we used the humoral responses measured in 6 different trials. So the 3 phase 1 trials that have previously been used by Pazin et al. So label EBL 1001 in red, 1003 in dark blue, and 1004 in light blue, in which the 44 subjects either come from Europe in red or from Africa in blue, and which were only followed during the first year after the first vaccination. And so in this plot, you can see the mean dynamics of the humoral response. So it's binding IgG concentration titled by ELISA essays. And so the dynamics over time in each trial, with a peak which can be observed 21 days after the second vaccination, followed by a slow biphasic decrease of the antibody concentration up to 1 year. And so in addition to the phase 1 trials, we also use data from 3 phase 2 trials. So labeled EBL 2001, gathering more than 70 European subjects, EBL 2002 with 137 African subjects, and 3001 gathering more than 2,030 subjects. And in this specific trial, subjects were followed up to 2 years. So at the end, we have approximately 500 European or African subjects followed between 1 and 2 years, which should at the end provide us more information than just considering phase 1 trials, and we should learn more information about longevity of the humoral response. So now I'm going to talk about the method that we applied on those data. So, so first of all, we use the mechanistic model that have previously been developed by Parza and al. So on the phase 1 data, which describes the humoral response over time after the second vaccination. And so in this structural model, we assume that antibodies can be produced by two populations of antibody secreting cells, which are called long-lived antibody secreting cells, labeled L, or short-lived antibody secreting cells, labeled S. And in this model, we have 5 parameters, Phi-L and Phi-S, which are respectively the influx of long-lived and short-lived antibody secreting cells, and delta L, delta S, their respective decay rates. And finally, we've got the delta AB, which is the decay rate of antibodies. And so in addition to this structural model, to account for inter-individual variability, a statistical model is considered, introducing a mixed effect model on each of the 5 parameters of the structural model with a fixed effect, which can potentially depend on covariates, and a random effect, which is assumed to be normally distributed. And so by fitting this model on phase 1 data, Parza and al. Identify a random effect on 3 parameters, Phi-L, Phi-S, and delta AB, as well as a covariate for the geographic region, so Europe versus Africa, on the parameter Phi-L, so the influx of long-lived antibody secreting cells, with, as we can see on this table, gathering the parameter estimates obtained on the phase 1 data. They obtain a higher influx of long-lived antibody secreting cells in European subjects than in African ones. And finally, the observation model, linking the observation and the structural model, is defined by the log10 transformation function of the antibody compartment, and we assumed a constant error model. So considering this model, the first part of the method that we applied on the data was to evaluate its quality of prediction by applying a 2-step approach. So the first step was to evaluate the robustness of the model estimated on phase 1 data. And so to do this, we only evaluated random effects using participants of phase 1 and phase 2 trials, using empirical bias estimates, so the EBEs. And in this part, we only considered data restricted to the first year of follow-up. So really to stay in the scope of applicability of the model developed by Parza and al. So we didn't consider any modification of the population parameters, of the random effect distribution, or of the error models. So in other words, we just applied the model estimated on phase 1 data on all our subjects, so to predict the humoral response for the approximately 500 subjects up to 1 year, and by only evaluating the random effects. We didn't re-estimate the model. And then the second part, so based on this estimation of this random effect estimation, we aimed at validating the quality of long-term prediction of the model, what we called the forecast, by predicting data from 1 to 2 years of follow-up, and then comparing them to the observations. So it was the first part of the work, which was about the evaluation of the quality of prediction of the model. And then the second part of the work consisted in updating our knowledge about the longevity of the long-term immune response using more subjects and longer follow-up. And so to do this, we use all the phase 1 and the phase 2 data. And first of all, we validated and updated the covariate structure in the statistical model. And in particular, we used several algorithms already implemented in the software monoliths, such as the algorithm Samba, SCM, and COSAC. And then we re-estimated, so once we updated the covariate structure, we re-estimated the model parameters, so using all the data. And finally, we validated the model process using a Monte Carlo cross-validation approach. So this was about the methods, and so now I'm going to talk about the results that we obtained. So first of all, the results about the validation of the model and the evaluation of its quality of prediction. So as mentioned before, we firstly evaluated the random effects and we predicted individual dynamics up to 1 year after the first vaccination. So which is represented by the blue parts of the individual plots. So each subplot represents the individual dynamics of the humoral response over time, where observations used to evaluate the random effects are represented by the blue dots. And model prediction is represented by the solid line, and the 95% prediction intervals by the blue shaded areas. And so we found that approximately 99% of observation fall within the 95% prediction intervals, showing really a good ability of the model to capture individual dynamics from the 6 trials, even if the model has only been estimated on data from the 3 phase 1 trials. And then, so based on this first part, so on the blue part, the model prediction, we predicted for each individual its long-term humoral response, so what we called forecast, represented so here by in each plot by the orange part. And we evaluated that approximately 90% of long-term observation, so meaning between 1 and 2 years after the first vaccination, which are represented by the orange circles, fall within the 95% prediction intervals, so within the orange shaded areas. And so based on those results, we can say that the model the model estimated only on phase 1 data and only on the yearly phase of the dynamics, so up to 1 year, has good quality of long-term prediction up to 2 years of the humoral response. And however, we can, even it's a good quality of prediction, we can notice that the estimation of the model can be improved. We obtain only 90% of the observation within the 95% prediction interval. And so, as I said previously, the second objective of the work was also, once we evaluated the good quality of prediction of the model, it was to update the model using all the new information provided by the additional data, meaning using more subjects and considering the longer time periods. So using all the data, we updated the value of the long-lived antibody secreting cells half-life, which is derived from the long-lived antibody secreting cells decade rate, so the parameter delta L. And so similarly to to the work done by Pazin and all on phase 1 data, if we add additional data, we still didn't have enough information on the new data to really estimate this parameter delta L, because we only have a follow-up up to 2 years. And consequently, we performed what we call a profile likelihood. So for different values of the long-lived antibody secreting cells, we fixed the parameter at different values, and we estimated the model for each value. And we calculated the log likelihood, which gave us this type of profile. And so based on this profile, we were able to update the value of the lower bound of the long-lived antibody secreting cells half-life around 15 years. So we cannot really estimate the parameter, but we can find a lower bound for the half-life. And we can see that this new value is, so we were able, using additional data, we were able to update this value, which was initially estimated at only 6 years using only phase 1 data. So using longer time periods, we were able to learn information on humoral response, long-term humoral response. And so then once we fix this, so the parameter delta L at the value found by the profile likelihood, we then updated the covariate model, so in the statistical model. And so by applying 3 automatic covariate model algorithm, which are so implemented in Monolix. So as I said earlier, the 3 algorithms are called Samba, SCM, and COSAC. So we tested 3 different algorithms really to validate the robustness of our results. And so for those 3 algorithms, we tested the addition of 7 potential covariates, which are continents, so Africa versus Europe, region, which stratify the Africa between East Africa and West Africa, the covariate related to the trial EBL 3001, and the covariates about sex, age, weight, and BMI. And we tested those different covariates on the 4 parameters, delta S, delta AB, phi L, and phi S. And so we just made one assumption. So the assumption, is that antibody decay rate is assumed to be non-region geographic, which means that the parameter delta AB cannot be adjusted for continent, region, and EBL 3001. And so then using the Bayesian information criteria, we were able to identify different covariates in our model. So first of all, we found an adjustment for continent, the covariate continent on the parameter PL, with a higher influx of long-lived antibody secreting cells in European subjects. So this is the same result that have been found on the previous study. So we validated the previous statistical model found on phase 1 data. And additionally, we also found a significantly higher decay rate of antibodies for men than for women. So women tend to have antibodies with a longer half-life than men. And finally, we also these results, even if we didn't found this kind of results in other study for Ebola, this type of results, a decay rate of antibodies higher for men than for women has already been found for other infectious diseases, in particular for SARS-CoV-2 in some studies. And finally, we also found a positive correlation between the parameters of delta AB and PL, meaning that subjects with a higher peak of antibodies will tend to have a faster decrease of antibodies after the peak compared to those with a lower peak. So this model, so we use this model as the optimal ones, and in this plot we represented the visual predictive check obtained for this specific model, which graphically represents the ability of the model to capture the central trends and the individual variability of the observed data. So in the blue and red shaded areas represent the 10th, 19th, and the median's prediction interval estimated on the data, which have been simulated by the model from multiple Monte Carlo simulation, while the solid blue lines represent the exact same percentile, but for observed data. And we can see that the model estimation are quite in accordance with the observations within each trial, and which shows the ability of the model really to capture the dynamics specific to each trial. Finally, so we performed a Monte Carlo cross-validation on the model with either 60, 70, or 80% of the subjects which were used for the training datasets, and so the rest for the testing datasets. And we particularly pay attention to keep exactly the same ratio within each trial. And for each percentage, we run 100 replicates, sorry, and we evaluated two criteria on the testing datasets: the root mean squared error and the percentage of coverage, meaning the percentage of data falling within the 95% confidence interval. And so in this table, we gathered the results that we obtained, and we can see that the results are quite stable for the three percentages, with a mean coverage percentage, so in the second line, which is higher than 95%, and the RMSE, which is globally lower than 0.1 log10 transform antibody concentration, which is consistent with the standard deviation of the error model of the model estimated on all the data, which was estimated at 0.1. So to conclude, on all those results, we validated finally the ODE-based model, which has been developed by Pazan all on phase 1 data, and its quality of prediction by considering additional data from phase 2 trials. And we also extended our knowledge about the long-term immunity with the update, in particular, of the lower bound of the long-lived antibody secreting cells, so from 6 years to 15 years. And through this work, we also developed an innovative pipeline of methodology that can be used for other infectious diseases. So, for example, COVID-19, by updating the model and providing more and more information as soon as new data are available. And however, we know that the model that we use is a very simple model, and that's why additional works are currently in progress in the team. So first of all, the improvement of the mechanistic model with the addition of memory cells compartments to be able to capture really the change of the immune response between the two vaccine injections, instead of just using data after the second vaccination. Yes, and the second work that is currently done on the team is the adaptation also of the model to capture the effect of a booster dose of AD2600. And so for the perspective, we would like to apply this type of model on SARS-CoV-2 data, in particular to evaluate the longevity of the antibody response, but also to investigate the impact of variants and to study the timing and the need for booster dose. And we are also working on a bigger model, incorporating not only the humoral response, but combining the modeling of the viral dynamics and the humoral response and their interaction in the immune system. And so just to conclude, I would like to acknowledge all the people that have been working with us on this project, so from Janssen Pharmaceutical and in my team, so Rodolphe, Mélanie, and Quentin. And I thank you for your attention.
Thanks, Marie. Thanks so much. I think we can start with the questions that are in the chat. So someone asked, why did you choose a normal distribution for your statistical model?
For the statistical model, you mean for the random effect? I mean, yes. So just a slide, just to be sure. Here, you mean here? It's because yeah, so we applied a log transformation on our parameters to ensure the positivity of the parameter, and so it's something usual to apply a normal distribution in this type of modeling. Yes, and if the question is a normal distribution for the observation model, it's because it's well known in the literature that the log-tape transformation of the antibody concentration is assumed to be normally distributed. I don't know if I answer well the question.
No, that's good.
Okay.
The second question is, how did you validate your model by using MCCV?
Yeah. Yes. Validated. Yeah. Actually, we performed MCCV just to verify that even if we select just a part of the subject, we still obtain our RMSE close to the standard deviation of the model and of the error model, and we just wanted to be sure that at least 95% of the observation falling within the confidence interval. I don't know if we could go further and use the Monte Carlo cross validation in a different way to validate the model, but we just wanted to verify that the property of the model was good in terms of prediction.
Great, thanks. And then the next one is, how did you estimate your random parameters?
The random parameters here. Yeah, okay. So to estimate the parameters, we use a monolith software, and it's and in monolith it's a maximum likelihood estimation which is implemented. So it's a SRM algorithm, and I don't know if it's a question. We use the SRM algorithm.
Okay. And why did you use BIC and not AIC?
Yeah, okay. For the covariate selection, I presume. Yes, so we use the BICC criteria because it's the only criteria that was implemented for the three algorithms, and we wanted to be able to compare the result obtained for the three algorithms. But we also tasted the ICC, the AIC. But with the AIC, we had not enough, how can I say that? There was too much covariates. We were able to select too much covariates with this criteria. It's not enough. Don't find the right word, but
it's broad.
Yes, it's enable too much covariates to be selected. So we use the BICC, which provide us enough covariates to have a good model, but not too much.
Mm-hmm. And so I had a question too. Can you go back to the individual fits?
Yes.
Yeah, that one. So your subject number 12. I'm interested in knowing, like, do you think that that individual actually did get immunity? Like, it's hard. So do you have one data point in the very beginning for subject 12?
Yeah.
Yeah, and so really this prediction for subject 12 is just some like a combination of all the other subjects.
Yeah, yes, that's right. Actually, when we look at this equation, yes, we take into account the mean dynamics within the population, and we were also able to estimate maybe a small proportion of the variation for this specific subject, because we got one data, but it's mostly the mean dynamics for this specific subject.
Okay. Thank you, Marie. I think that there will be some more questions during the discussion session.
Okay, thank you.
And so we'll move on to our final